Mics are open.
Good to go.
Just waiting on the live stream. It's almost there. A couple seconds. Okay. Okay, hello everyone, and welcome to Concordia University's Fourth Space, where we're taking a closer look behind closed doors and into spaces and projects you might not know about at Concordia. And today, Fourth Space takes a closer look at the MON Lab, which stands for Metabolism, Obesity, and Nutrition. And we'll be touring that with public scholar Mohammed Ilyas. But just to help situate you, we are streaming to YouTube, we are live from Fourth Space, and we are located on unceded Indigenous lands in Tiohtià:ke, Montreal. Here at Fourth Space, we work with our university community to help mobilize knowledge by co-creating daily activities that examine research questions and projects and things happening across the university. And we're hosting this event live on location. So some things can happen, connectivity issues are to be expected, but please stick with us as we show you around. We're also running this event as a live stream Zoom meeting. So anyone joining us there, if you have any questions, pop those into the chat. We'll try and get those over to the space if we can. And same for anyone joining us here in the space, just raise your hand. We'll get a microphone over to you. All right, let's go. Mohammed Ilyas, could you introduce yourself? Could you tell us where we are today?
Hi, Ed. Thank you so much. So my name is Mohammed Ilyas, and I'm a PhD candidate and a public scholar in the Department of Exercise and Health Sciences. So yes, we are at the School of Health building at the Loyla campus, and we are at the second floor, and I'm going to show you the MON Lab. Let's get in.
Great.
Oops.
I
Card
feel
isn't
like
working.
this is lab life right here. We're witnessing it.
Sorry for the interruption. I don't know, at the last minute they cut my access card. So let's get in.
All right. Hello. Now we're just going to take a look around.
Ace is just going to take you on a quick tour, Doug, and then we'll explain kind of everything that you're seeing as she goes.
Thanks, Birdie. I'm just looking around with this.
We've got lots of students hard at work.
Yeah.
And to explain, maybe.
Yes, thank you. Thank you so much. So this is our MON Lab. And before I talk about the MON Lab, I would like to introduce my professor. So here is Dr. Sylvia Santosa. So would you like to talk about the research and what is happening here, please?
Hi, Sylvia.
Hi. Nice to see everyone. Welcome to our lab. It's the Metabolism, Obesity, and Nutrition Lab. Here in the lab, what we do is we want to try and understand how fat tissue from different areas of the body affect our overall health, as well as what affects those fat tissues. So whether you're, for example, male or female, how does the fat tissue in the stomach area affect our overall health differently? And what are those factors that really are affecting our health in that tissue?
Thank you, Dr. Santosa. So I will be talking about the MON Lab, and thanks for joining us.
Okay.
And, yeah. So
Thanks.
I'll take over.
Thanks, Sylvia. It's great to hear it. Yeah.
Thank you.
Thank you, Douglas. Yes, so this is the MON Lab. So here we use a bunch of different techniques, like including the physiological, nutritional, and biological techniques to understand that every individual is unique, in terms of their fat depositions. And we want to see that if they are gone through the bariatric surgery or interventions, diet intervention, and how their body responds to that. So my project is about the characterization of adipose tissue in the individuals with type 2 diabetes. So what we're going to do is actually we usually send an invitation to the individuals around the Montreal area to invite them to participate in our study. So once they respond back, we invite them for the biopsies. So first I want to show you the biopsy trays. So you can see there here are a lot of tools which we use during the biopsy procedure. It's a mini invasive surgical procedures where physicians comes and they actually perform the biopsies on the individual. You can see the different sizes of the needles, and you can see the syringes. Then we have the anesthetic, lidocaines. So all this stuff is actually used to collect the adipose tissue. And the most important one are these needles. So these are the biopsy needles which are being used to actually aspirate the fat from individuals. And after that, once we collect that fat, and actually we bring it to the lab and process it here.
Mohammed, are people volunteers for this, or are they recommended? How do people participate in that program?
So basically, we have a collaboration with multiple different institutions, mainly because we are inviting the people who are diagnosed with type 2 diabetes and obesity. So that's why we actually send an invitation to the hospital, and from the hospital, we actually invite them, and they come over here in the School of Health building, and then we perform the procedures on them.
Okay, great.
Yep.
Wonderful. Okay. So once we've collected the samples, do you want to take us over
exactly.
to the next step?
Once we collect the tissues, so we actually bring them to the lab, and we want to process it as soon as possible because those are the live cells are there in those tissues. So we want to preserve those cells and do the experiments for the later. So the first area which I'm going to explore is actually the cell culture area. So come with me. Silence. So here is the cell culture area. So we bring up the adipose tissue here, and after the digestion, we actually process the samples here in this biosafety cabinet, and we actually grow cells, and the cells multiply. And after that, when we have the enough cells, we actually store them in the freezer or the liquid nitrogen for the later use for the long-term treatment options.
But the cells you get from the sample, Mohammed Ilyas, you have to act quickly. You do that right away.
Yes, we have to act quickly, not right away immediately, because we have to finish the biopsy, comes to the lab and processes immediately. But once we freeze them, they are freezed in the liquid nitrogen. I'm going to show you later on. And liquid nitrogen actually preserve the integrity of those cells for the later treatment options or whatever the experiment we want to do on those cells.
Thank you.
Welcome. So here's the Alex. She is doing some experiments, and she's a second-year PhD student working on the project, understanding what is the interaction of adipose or fat cells and the cancer cells. Thank you.
Wonderful.
Wonderful. Okay. So this is the second step, and after that, coming on to the next stage, once we have digested the tissue, we actually take the digestion fluid or liquid under the microscope to capture the images so that we can understand the different sizes of adipose cells. So adipocytes are here. You can see the image here. Once we capture the image, so you can see on the image there are different sizes of the adipocytes. There are some bigger, there are smaller. So these actually are unique for each individual. So it is one of the characteristics where we actually identify that how the cell sizes differ from person to person. And later on we analyze it for the comparison purposes. So here's the Kayla. She's working on cell sizing. She's also a PhD student here. Thank you, Kayla.
Thank you.
Moving on. Thanks, Doug. So these are the freezers and the fridge where we store the reagents, which we use to process all the biological samples. You can see different types of reagents are here. So just to mention that MON Lab is one of the kind lab in Montreal where we process the human samples, which are very precious, or it's hard to collect because nobody simply agree to give away their tissues, right? So because they are surgical procedures. Yep. So we are in the next area. So this is the shared area, which is shared between our lab and the other researchers at the Health and Exercise Department. So our next station is after having the cell sizing, we have some of the plasma and serum extracted from the blood samples. So here is the Anjali. She is also the second-year PhD student. Hi, Anjali. So she's working on the ELISA. So let me tell you, the ELISA is a kind of technique. It's a enzyme-linked immunoabsorbent assay, which we use to actually detect the biomarkers and different hormones and proteins to understand that how it alters from person to person. So this is also one of the characteristic. So since I mentioned earlier that my project is characterization of adipose tissue, so this is also one of the characteristics where we understand that in each individual, the characteristics vary in terms of biomarkers as well. And our participants, they go under the bariatric surgery and they have the weight loss. After weight loss also, we measure again their protein expressions and understand that how much is the difference after the weight loss.
And that can change from person to person also.
Exactly. So this is a microplate reader machine. So we have the ELISA plate, which is actually then being, processed here, and then we can read the results on the computer, and it shows us the optical density because the light passes through those plates and identify based on the concentration of different biomarkers. Okay. Thank you. So we have another station here, which is Western blot station. So Frank is working here. Frank is highly qualified and one of our lab technician, and he's helping all of our projects. And you can see that he's running the gel here and pouring the samples inside the gel. Hi, Frank. We are not disturbing, hopefully.
Yeah.
So really focused. So yes, so the Western blot is also one of the technique which is used to see the protein expression from the tissues, adipose tissues, fat tissues, right? And that one is also used to identify the functions of cells, especially the fat cells and muscle cells. Yeah. Thank you, Frank.
Wonderful. So we've collected the cells, we've put them through a few different tests to experiment, and then what comes next?
Yes, there is another actually characteristic of adipose tissue where we can identify the immune profile of the individuals, immune profile of those collected fat tissues. So that is also tells us about the inflammation and overall inflammatory health of that particular individuals. So when we collect the fat sample, we digested it, we lysed it, we break down the all small molecules in cells, and then we separate the cells which are interest of us, which we are interested to study. Those are immune cells, could be lymphocyte, macrophages, T cells, B cells. And then there's a gold standard method we are using to quantify those cells. So I'm going to show you. Come on.
All right.
Here we go. So this is flow cytometer. So this is the machine which actually help us to count the cells. So it uses the different types of lasers, red, violet, blue lasers. So those lasers actually identify the cells based on the staining of the antibodies colors. So we usually take the cells, we extracted them, we stain them with the antibody markers, which identify that what type of cell it is. Okay. So once we feed the tubes, fax tubes, so it has the samples or extracted cells with the staining antibodies, we feed it to the flow cytometry, and then we process it. When we process it, we get the graph. So I'm going to show you the graph here. If you look at this graph, it has a, scattered cells populations, distinguished cells populations. So if you look here, there is a side scattered and the forward scattered. So side scatters actually helps us to identify the, disperse the cells based on their size, okay? And the forward scatters actually identify the cells based on their granularity. So based on their granularity, we can see that there are distinct populations. This is one population, which is your macrophages. These are lymphocytes, then there are monocytes. So that's how we identify the different types of cells. And each cell has a particular role in, towards the body response towards a certain disease like type 2 diabetes and obesity. So this is the end point where I actually understand the inflammation, status of that person based on their immune profile. So the bunch of different techniques are being used in this lab to understand the characteristics of adipose tissue in individuals who are living with obesity and type 2 diabetes. Yep. So let's move and let me show you some other stuff here in the lab. Yep. So here is Claire. She is our postdoctoral, very hardworking. Hi, Claire. Say hi to camera.
Hi.
So she's also helping us and mentoring all the students here. So this is also one of the area of the lab where we process the samples. This is called as mRNA room. So where we extract the messenger RNA from the cells or tissues, and then we run the gene expression studies. Yep.
Okay.
Wow, there's a lot going on in here.
Yes, so you can see here. So the next ones are the liquid nitrogen tanks.
Okay.
So these liquid nitrogen tanks are very rare in, I mean, not every lab has these liquid nitrogen tanks. So this has a liquid nitrogen inside, which is very, we can say that low temperature, really low temperature it has.
Right.
And why we use it to store those samples? Because it's maintain the integrity of those biological samples. Because the biological samples and the human samples, they are very, very precious. It's very hard to recruit individuals and convince them to donate their fat and tissues, you know. And
Right.
we are thankful to all of our participants. Yeah, let's not forget them actually. So they have actually donated those tissues, and we process them, and we got the cells, we grow the cells, we apply a lot of different techniques to run different experiments, and then finally we store the samples here. So let me see. So you can see this is kind of, you know, fumes are coming out.
Whoa.
So these are the liquid nitrogen tanks. Okay, so moving on, we have another room here. So this room, you can see that we have the deep freezers, you know, these are the -80 freezers. So you can come in.
Minus.
So our lab is very unique lab where we have 3 of them, because these are very expensive to have them, you know, and we have 3 of them here. So to store our samples, let me open. So you can see there are a lot of different files which are having a lot of different samples which are being processed. Okay, at -80.
At
So
minus 80.
I'm going to keep it back.
Yes, that's right, Doug. -80. You heard that right.
And how long do you keep the?
Today.
It's colder than Canada, right?
It's called, yeah, we found
Yeah,
it's called-
almost a little bit.
Canada, yes. Thank you,
Yeah.
thank you, thank you. So that's also, we are almost done with our lab tour, and that's all from my side. So just to mention that in
Yeah.
this lab, we conduct a lot of different experiment based on individual products. So we are actually running the projects which compare the protein, animal-based and plant-based proteins comparison and interventions, and understand the effects of them on the inflammatory profile of the individuals living with obesity. And for my projects, for the characterization of adipose tissue, so we are collecting the tissue from 2 different regions, abdomen and the thigh. And then after that, we are applying the techniques to understand the characteristic and compare those characteristics, and how this knowledge is going to help us. This knowledge is going to help us to find the solutions, treatment solutions, and the better interventional approaches for the individuals who are living with obesity and type 2 diabetes. And we're going to have the healthier life for those individuals. And thanks to those who participated in this study. Is there any
Absolutely.
questions? Please ask.
Mohammed Ilyas, one question I usually like to ask is how someone got into this type of research, but I know you have actually a quite diverse background, but I am still curious how you came to this particular project.
Yes, so the thing is, my undergrad was physiotherapy. So I am a basically physiotherapist, and I work for individuals to make their life healthier in terms of their physical activity and overall conditioning. But when I did my master's, so I was curious in the clinical practice that how these diseases actually, what is happening at the molecular basis, you know, at the ground level. I want to know that what's going on in the human body. So that actually initiated interest in me to go and pursue the research in molecular medicine. So I did my master from Malaysia, and then I tried to, work on the people who are living with obesity and work on the genes. And after that, when coming to the Canada, I found this lab, which is very unique because they're working on the clinical samples, and it's very rare to find the clinical samples and store them and work them. So this lab is kind of interest for me to understand more molecular mechanisms and understand the more etiology of type 2 diabetes, and then find those mechanistic pathways where we can have the target approaches to give a better treatment. So that's why I wanted to be a clinical scientist and finally work as a clinician and also the scientist as well. So that's the whole.
That's really great to hear. Also, just the spectrum of going from general wellness and physical activity all down to a very specific set of questions that also generally affects health. That's a really interesting pathway.
Yes.
I know, Mohammed Ilyas, thank you so much. I know we've only just scratched the surface of what's happening at the MON Lab, but I really appreciate your time showing us around. I'm just going to let everyone know we're going to close up from here, but I wanted to thank you again for your time, and thank you for all of your colleagues to participate and show us what's happening. Team Fourth Space, Birdie and Isa, thank you to you also.
We have one question. Yeah.
Oh, we do.
Isa has a question, I think.
Okay, great.
Can you guys hear me?
I can hear you. Yeah, go ahead.
Okay, I'm trying to hold the camera at the same time.
Okay.
So when you want to come up with these treatments, what does those treatments look like? Do they look like medications? Are those injections? Are those kind of like better ways to treat obesity and diabetes with lifestyle changes? What does that mean, and what have we like come up with so far?
So it's a very good question, actually. So the treatment varies. There are a lot of different management strategies being used in the market to manage these diseases like obesity and type 2 diabetes. So yes, we have 3 to 4 different projects understanding the different treatment approaches. The one is actually understanding the diet intervention. So it could be the lifestyle modification. It could be the dietary intervention. One of that project is exploring that kind of intervention. And then there is another project which is mine to understand the characteristics. So we are working on the physiological and pathological pathways, and we want to identify the potential targets where we can, where we can actually apply the pharmacological interventions to treat the cells and see how they respond to certain treatment. So that's why this project is focusing on that kind of intervention. Then we have another intervention where we are actually trying to understand that how exercise and the strength training actually helps to improve the outcomes of those people who are living with obesity and type 2 diabetes, and they are undergoing the bariatric surgery. So I can say that in our lab, we are exploring all 3 or 4 different horizons of treatment options by getting the basic knowledge from doing these experiments. I hope I have answered your question. Yep. Thank you.
Wonderful. Like you said, Doug, we clearly have only scratched the surface at what's happening here today.
Yep.
Seriously. Okay, well, we're going to say goodbye here from Fourth Space. Mohammed Ilyas again. Thank you.
Thank you, everybody.
Thank you, everyone. And we'll just let you know, we'll be closing up the live stream and the Zoom now, but this tour remains available on our YouTube channel, so feel free to, and I encourage you to like it and share it, etc. And we'll see you again soon. Have a great weekend.